Welcome to Extreme Genes, brought to you by The Church of Jesus Christ of Latter-day Saints. What happens to your spit after you send in that sample to the DNA companies? Hi, it's Fisher, and I'll be talking to Paul Woodbury, a DNA specialist with Legacy Tree Genealogists, about spit to screen. Just how that magic happens that gets you the results you're looking for in ethnicity and DNA matches. Plus, we've got Team Green on from the latest season of Relative Race, talking about an amazing experience and perhaps a relative race first. That's this week on Extreme Genes Family History Radio, brought to you by FamilySearch.org. Discover, gather, connect. A presentation of the Church of Jesus Christ of Latter-day Saints. And welcome, Genies, to America's Family History Show, Extreme Genes and ExtremeGenes.com. It is Fisher here, your radio root sleuth, on the program where we shake your family tree and watch the nuts fall out. And this episode's brought to you by BYU TV's Relative Race, Sunday nights at 8 o'clock Eastern, 5 o'clock Pacific, and they're coming down to it. What a season it has been. And what a show it is going to be for you today. We've got a lot of DNA to focus on today, and I'm really excited to get Paul Woodbury back on. You know, he is the DNA specialist over at Legacy Tree Genealogists, one of our great sponsors. And Paul's going to talk to us about how your spit gets to the screen. You know, when you do these DNA tests, what happens from the time you spit in that little tube to the time you get your ethnicity results and your DNA matches. He'll go through the whole thing with us, and I know it's going to be absolutely incredible. Hey, if you haven't signed up for our weekly Genie newsletter yet, please make sure you take care of that. It's absolutely free. Once a week, you get a blog from me, plus links to past and present shows and links to stories that you, as a family historian/genealogist, will appreciate. Hey, let's head out now to Boston and talk to my good friend David Allen Lambert. He is the chief genealogist of the New England Historic Genealogical Society and AmericanAncestors.org. How are you, David?
Hey, I'm doing good after flapping my wings and finally getting back home after 3 weeks of travel.
Boy, you were all over the place. That's true. By the way, I got to give a shout out to some folks there at our anchor station in New England, AM 680 WRKO, the voice of Boston, right where you are. They raised more than $230,000 on Veterans Day to benefit the Disabled American Veterans Department of Massachusetts. And they did this during a radiothon that went between 6 in the morning till 8 at night. So congratulations to Jared Diglio and the crew and our anchor station in New England, AM 680 WRKO, the voice of Boston. Love that.
Yeah, great station. We love it out here very, very much. And I love it even more that we're on it.
Yes.
So we have now lost what may have been the last Pearl Harbor survivor from Massachusetts, George Hersey, age 98, who, when he retired at 83, was the oldest school bus driver in America.
Oh, wow.
He lived in the town next door to me. On another sense of veteran news, the Vietnam War, back 50 years ago, there were so many vets that fathered children. And the great thing is that DNA is reuniting many of these babies with their families. So this story has to do with Kirk Kellerhals, a 50-year-old from Norfolk, Virginia, who was born in Vietnam to an American father and a Vietnamese mother. He had thought his parents were dead. He has now been reunited with them and had got the whole story.
Yeah, and they're all here in the United States. Yeah, as a baby, he came over here because her parents were horrified that she would have a baby with an American soldier. So she was separated from the soldier, from the baby, and there were documents that said both of them were dead. So it wasn't just assumed they were dead. As far as the records went, they were. But the DNA test found the mom, and she had been back in touch with the soldier, and now they've reunited, and it's just, he says it's just the most mind-blowing experience of his life.
Be able to reunite with him is amazing.
Yep.
Thank you, DNA. Well, you know, the story we talked about earlier, another veteran story, this one goes back to World War II to an American GI that was actually in a prisoner of war camp who, to relieve the stress and the drudgery of being a prisoner of war, made a comic strip. Why don't you tell the story? Because I love this one.
Yeah, his name is Howard Weisling, and he was hungry, he was homesick, he's in Barth, Germany at a POW camp. And he drew a comic book on cigarette wrappers bound together with scrap metal. And then they took this thing and they sent it around the camp, one panel at a time. And the soldiers, according to Howard's son, Morgan, just looked forward to it all the time. It gave them something to anticipate. So when the camp was liberated, the book was left behind, but somebody wound up getting it. And somebody now has tracked down Morgan, Howard's son, and gotten it back to him. And Morgan is saying, it's just like getting my father back again. It was like him being able to tell me the story all over again, except this time it was real and in my hands. Isn't that incredible?
It really is, and you just never know what's out there. Amazing value to the family when you can reconnect the piece like that.
Right.
You know, sometimes when we're looking for things, like I went mudlarking in the Thames in London and I found all these pipe stems, but Bob Mukel, an anthropology instructor at Capilano University, was in the backwoods of British Columbia and stumbled upon what was a Japanese town, now a Japanese ghost town, where these road workers worked from the 1920s to 1942, and he's found a plethora of artifacts that really tell the whole story of them being there at this camp.
Yeah, and apparently they had the same situation in Canada as we had here in the United States. And ultimately, these Japanese Canadians were rounded up and interned. And they say that the evidence there is that everything was left behind very quickly. So that's why they think they were rounded up, but living in the middle of the forest all this time just to avoid the persecution.
This included, of course, women and children as well as men.
Yeah, absolutely.
Well, you know, Christmas and the holidays are getting close, and I want to thank Shamrock genealogist Melanie McComb, who we have on the show. She gave me an interesting news story. So I'm on my Christmas list now, and I think you'll agree. Ground-penetrating radar. What? It's a must-have.
What?
Yeah, well, you know, so you can find those hidden cemeteries, which is what's going on at King High School down in Tampa, Florida, where they're trying to locate the 250 or so graves that were part of the pauper cemetery of Ridgewood Cemetery that were never moved. Seems like a scene out of Poltergeist.
Ooh.
They're out on the 50-yard line of the football field looking around for an actual cemetery.
Oh, no.
You can see bleachers in the background, so I don't know.
So that basically they were playing football games over the top of these people? Oh.
So around the school area, yeah, because you can see the bleachers in the background. So it's interesting. So, yes, Santa Claus, all I want for Christmas is ground-penetrating radar.
Nothing says Christmas like GPR. All right, David, thank you so much. And, of course, you're coming back for another Ask Us Anything at the back end of the show, so we look forward to that. And coming up next, we're going to talk to Paul Woodbury. He is a DNA specialist at Legacy Tree Genealogist, and he's going to take us from spit to screen. What happens with your saliva once you fill that tube before it gets to the screen? And it's always a pleasure to have my good friend Paul Woodbury on the show from Legacy Tree Genealogist. He is the DNA specialist. And Paul, I know you've been working on an interesting explanation for people to understand what's going on, because it is really kind of odd, isn't it, that we can spit in a little tube and all of a sudden our genetic world is open to us. Matches and potential breakthroughs. I just had one myself on some third-grade grandparents after 35 years because of a bunch of fourth cousins that started showing up, matching many of us who descended from my second-great grandfather. I mean, it's amazing stuff. So let's talk a little about this idea of from spit to screen. What's all going on in there? How you doing, man?
I'm doing great. Thanks so much for having me, Fish. So, like you said, it's a little bit confusing for a lot of us when we spit into this tube, or maybe we swab the inside of our cheek, depending on the company you're using, and we send it into a lab, and suddenly it comes back to us with an ethnicity estimate, with a list of matches. And what are the steps that in between where you spit and you get that report? And there's a lot of science and technology that's going on behind the scenes, and it can get a little bit complicated, but I'm going to try and distill down some of the most basic principles of what is happening with your DNA between those two steps.
Oh, boy, here comes Mr. Science. Put on your glasses and your pocket protectors. All right.
Yep. So first, DNA is in every cell of our body.
Yep.
We've got a complete copy of our DNA in each cell. And each cell differs from other cells because they express or they read different parts of that DNA. It's kind of like a big factory. Your body is a huge factory, and you've got different departments in that factory.
It's a department store.
Yeah, it's a department store.
There we go.
There we go. And and each department, has a copy of the employee handbook. But if you're in the, in the cashier department, you're only going to be reading the sections that refer to you as a cashier. And if you're
Sure.
in the if you're in management, then you're going to be reading perhaps a little bit more of the handbook. And so that's kind of the way that our cells work. Our cells are different from each other, and they perform different functions because they read different parts of that handbook.
Okay. But they all have the whole handbook?
They all have the whole handbook, each of our 30 trillion cells in our body.
Oh, wow.
So what are the cells that we are actually looking at with these genetic genealogy tests? There are two main types depending on the type of test that you take, and which is kind of surprising considering that both of the types of tests are from your mouth.
Right. Yeah.
But when you spit, you actually get up to 60 or 70% of the cells that they're looking at, the DNA that they're grabbing from those cells, are coming from white blood cells. And if you do a cheek swab, most of the DNA that they are obtaining from a cheek swab actually comes from your cheek cells. So there's a little bit of a different source that you get from those two types of tests.
Sure.
Something to keep in mind, and something interesting, is if you have a blood transfusion, then you might want to wait a few months before taking a DNA test because those white blood cells will be from your donor rather than your own DNA.
Yeah, and there was a story recently, too, wasn't there, about umbilical cord blood that got into somebody and then became part of their DNA, and they matched that person like 25 years later.
Oh, interesting. I had not heard.
Yeah,
that
it's
one,
very
but,
recent,
yeah.
very recent story, and so they're all singing "Kumbaya" like their family. It's really interesting.
Oh, wow. So yeah, you want to be careful if you have a blood transfusion recently. If it's been a while, then usually your own DNA is replaced in that blood. On the other hand, if you have a bone marrow transplant or stem cell transplant for bone marrow, then you really want to be careful on the conclusions you draw on your DNA test results, because the DNA from that bone marrow donor is going to be represented in any sample that you take.
Oh, wow. For how long?
That's permanent.
Really? So it basically changes your makeup to the person that donated.
It changes the blood, and because we get a lot of those white blood cells as part of the test,
right?
That is often what will show up, and sometimes it will result in a failed run because they're seeing your DNA and your donor's DNA
at the same time.
And so that just messes them up and says, "What's going on here? You have two genomes," or it may be possible that you will get a good sample, but there's no knowing, is it your donor's DNA or is it your own
right. DNA? And you don't want to get arrested for a crime that the donor committed, right?
Right.
Yeah.
So those are some things to keep in mind. Well, so once you take this test, you spit or you rub the inside of your cheek, you put it in the tube, and you isolate it in this solution that they provide as part of the kit. And that keeps your DNA stable for the time period from when you take the test to when it arrives in the lab. And once it arrives in the lab, they add a detergent, kind of like a soap. And the reason for that is that way they can break open the cells that have been isolated in that sample. Because cells actually, each cell has a membrane around it, and it's called the lipid bilayer. And really, it's just two layers of fat that separate the cell from all of its surroundings.
Oh, I got more than that.
And when you add dish soap to a greasy pan, you note that the soap kind of gathers around that grease.
Sure.
And it isolates the grease, and it makes it possible so that the grease can mix with the water. And that's exactly what detergents do with our cells. They come around those fat layers around our cells, and they break them open, and they release all of the stuff inside the cell, including your DNA, into open mixture.
This is amazing.
Isn't that awesome?
Yeah,
so,
I had no idea.
So that's what they add soap, they add a detergent, and they break open the cells. Well, what happens when they open up the cells, there's some problems that we can run into, because cells don't like to have your DNA just running around wild out in the open. In your cell, the DNA is always focused and it's always centered in the nucleus. Unless it's mitochondrial DNA, in which case it's in the mitochondria, but it's always in its proper place. And so if your cell breaks open and there's DNA floating around, the cell actually has proteins that have a specific job of destroying DNA that is gone awry.
Runaway DNA.
Runaway DNA.
Rogue DNA. Crazy.
Rogue DNA, there are proteins in your cell that their job is to destroy runaway DNA.
Wow.
So to prevent that from happening, they add some chemicals that actually destroy the proteins but leave the DNA intact.
Oh, wow. So it kills the killers.
It kills the killers. And then they add salt to make all of the debris from the cell, from all of the tissues, all of the different things, perhaps some of the food that you ate before you took your DNA test. And it makes it all clump together, and then they spin it at hundreds of cycles per minute so that it can get rid of all of that debris, and it leaves the DNA in the solution, in the liquid part of this mixture.
Amazing.
Once they do that, then they add alcohol to make the DNA clump together, and they spin it again and isolate the DNA. So really, from this step of testing your DNA, the first step that they want to do is they want to isolate the DNA from everything else that's in that sample, all of the other things that are in the cell, all the proteins, all of the fats, all of different things, and they just want to get at the DNA.
That's amazing. Wow. I had no idea.
Isn't that amazing? And
Yeah.
so once you isolate the DNA, in order to analyze your DNA, you have to have a lot of copies of DNA. And usually you need a lot more than is provided in just a single sample. So the next step that they take, this first step was isolating the DNA. The next step is they copy that DNA, and they copy it, and they copy it. And so they copy it so many times, up to thousands of copies of your DNA.
Now, wait a minute. Are you talking physically creating copies, or are you talking about digitally creating copies?
Physically creating copies. So what they do is they isolate the DNA, and they add proteins, and they alter the environment to encourage the DNA to replicate itself, which is a natural process that happens in our bodies. Every time a cell divides, it creates another copy of the DNA. So they replicate that environment, and they speed it up so that you can create potentially up to billions of copies of a particular DNA segment within just about 4 hours.
Shut up. 4
Starting
hours?
with just one segment, just one copy of the DNA, you can go to billions of copies within a few hours.
That blows my mind.
Isn't that amazing? So that step is called amplification. And what they're doing is they're just trying to make as much DNA as possible so that they can perform some really strong analysis on it.
And they do. We're talking spit to screen with Paul Woodbury, the DNA specialist at Legacy Tree Genealogists. And I've never heard any of this stuff before, Paul. All I know is I spit in a tube, I wrap it up, I make sure it's preserved, I stick it in a package, I stick it in the mail, and the next thing I know, I've got matches. You're telling me there's a lot of work involved in this.
There is, and we're only about halfway there.
Paul, you've been taking us on a world of science. This is like 5th grade all over again. I don't have any goggles on or anything, but you're taking us from spit to screen. And when we last left our spit, as I recall, we had opened it up. We're now replicating it into billions and billions of pieces of DNA. Pick it up from there.
All right. So, we started with isolating that DNA, trying to get it out of all of the junk that was also available in your sample, and then replicating it so we have lots and lots and lots of copies of your DNA to be able to analyze.
And why do we need that many?
So I'll tell you why, because of the technology that we're using.
Okay.
And this is where it gets a little bit different, depending on the type of test that you're taking. Y-DNA, mitochondrial DNA, or autosomal DNA, they're all going to be a little bit different in how we analyze them and how we determine the values and provide the results related to those tests. But since most of us probably have done autosomal, or at least are most familiar with the autosomal tests from the different companies, I'm going to focus in on autosomal. and talk a little bit about how we analyze that autosomal DNA. First, it's important to recognize that our autosomal DNA is composed of billions of base pairs of DNA. So DNA is composed of 4 bases, what we call A, T, G, and C. And A will pair with T, and G will pair with C, and that's why we call them base pairs.
Okay.
Okay? And in the DNA that we carry in each cell of our body, we have that DNA organized into 23 pairs of chromosomes, 1 set that we get from mom, 1 set that we get from dad. And in total, we have about 6.2 billion base pairs
Oh, wow.
in our DNA. So when the companies test your DNA, they are not looking at all of those 6.2 billion base pairs because, surprising fact, all humans share 99% of their DNA with all other humans. And so looking at every base pair would be kind of useless because most of it is going to be the same across everybody. So what they are actually looking at, they're looking at hotspots for variation between people. And the type of variation that we're looking at with autosomal DNA tests is called a SNP. It's a type of mutation in your DNA where one base changes to another. Perhaps originally the DNA was TAAG, but then a mutation happens and suddenly it's TATG. An A changed to a T.
Okay.
Okay? And so what we're looking for with SNPs is we're looking for variations that are common enough in the population. So these mutations have happened long enough ago that they are spread out across the entire population. And in our example, some people will have A at a particular location. And keep in mind, we get 2 copies of our DNA, one from mom and one from dad. So they might have an A from mom and an A from dad. They might have an A from one parent and a T from the other parent, or they could have a T from mom and a T from dad. And so what we're looking at these locations where we have variation is we're trying to figure out what are the values at those locations so that we can find out what values you share with other people and with other ethnicities.
Oh, wow. So we're talking about the ethnicity side of it or the matches or both?
So they use this SNP analysis for both matching and for ethnicity. But they look at it a little bit different in order to get those 2 different results.
Because you still have to use matching for the ethnicity, right? I mean, you have to compare it to a population base.
Yeah, so we're using this analysis for both of them. Well, what the companies do is they look at about between 500,000 and 700,000 of these locations that are hotspots for variation in the population. And the way that they do that is each of the companies use a company called Illumina, which produces this technology that are called SNP chips. And remember, SNPs are the variations that we're looking at.
Sure.
And what they do with these SNP chips, these chips are just glass plates, and they have thousands of tiny microscopic silicon beads attached to these plates.
Wow.
And these silicon beads, each of them has a manufactured probe. It's kind of like a little piece of Velcro that's sticking up from this bead. And what we do is we cut up the DNA and we send it across this plate, and your DNA will stick to these little probes. It's like you're sending your DNA through a field of burrs. And
oh my gosh!
It connects to those burrs, and it's very specific in the areas that it's connecting to, and that's why we need a lot of copies of your DNA.
That makes sense. Sure.
Right? We need lots of copies of your DNA so that we increase the chances of your DNA finding its complement on this glass plate.
Wow.
So once the DNA sticks to these little beads, we introduce some fluorescent A's, T's, G's, and C's. And the probes are designed so that they stop just short of the variation point of the SNP. And so once we introduce these altered bases, those bases will attach to your DNA and will be complementary to what the value is at that location. Next, we apply lasers to it, and we let the DNA fluoresce, and the computer will analyze the color of those fluorescents. It knows exactly what...
Who thought this stuff up, Paul? Who thought this up? This is amazing.
Isn't it amazing?
Yeah.
And so each bead has a known location, which corresponds to a known location in your genome.
Crazy.
And so based on the color that comes out of that location, the computer can interpret what the values are that you share at that location. If it's green, it's TT. If it's red, it is AA. If it's yellow, then it's TA. And so then it creates a raw data file, and that's what the companies then apply their algorithms to, and they compare that raw data file to known populations to find the incidence of different SNPs in different worldwide populations, and that's how you get your ethnicity result.
Okay.
They also compare your raw data file against everyone else's raw data files, and if they find a long line of common variations where you share the same variations over, say, 500 to 600 consecutive SNPs, then that means you share a common ancestor recently, and they can use that to estimate how closely related you are to those other people.
Wow.
And that, my friends, is how we get from spit to screen.
That is insane. You got to just hand it to the nerds that we used to look down on so much in high school, right? That they were able to put this whole thing together. I know it's way over my head. All I care about is I spit, I send it in, and yes, the results come up on the screen. And the amazing thing about it is how life-changing it all is, right?
Absolutely.
You consider all the people who have made discoveries that they never imagined they could, and I think many of us are included in that. Paul, thanks so much for the explanation. My head's still spinning. I'm going to have to listen back to this a couple of times,
Sounds good.
See if I can figure that out. Although I don't think I'm going to be sharing much of this with anybody else personally, okay?
Sounds good.
He's Paul Woodbury from Legacy Tree Genealogists. He's the DNA specialist there. Thanks so much, Paul.
Thank you.
Now, always exciting to get to visit and get the inside scoop from our teams on Relative Race, BYU TV's great show. And we've got Team Green this week. We're talking to Deshay and Chris, and welcome you guys. It's just been a joy to watch. And Deshay, this past 2 weeks have been insane for you. Take us through this.
Wow, hello. Thanks for having us. Yeah, the last 2 weeks have been doozies. I mean, my whole journey leading up to ultimately meeting my biological father, Mike, and not a whole lot of information out there on my biological mother after day 7. And so to be just hit with that on day 8, it was like, I mean, I was speechless that day, and you saw that, I think, on that episode, but I just was not expecting it. It was amazing. I mean, very few teams in the, I think, history of the show have been able to meet both parents,
right,
and even a grandparent. Like, I don't know how many teams have been able to do that, so I feel blessed and fortunate to have that complete puzzle. It's amazing.
Yeah. And you got your answers. You know, that's the thing this season on Relative Race is everybody has somebody on their team looking for a birth parent. I know, Chris, you've been really supportive, but you had a moment also meeting the second cousin where the roles were kind of reversed in this show, right?
I did. Knowing that I could meet some relatives that I didn't know was an interesting prospect for me. But when I met Becky, it was amazing to begin with, but finding out her story that she was adopted and, in a sense, I was the person on the inside of the door, being the first biological relative that she met was a really huge role reversal and amazing and exciting, and I felt blessed to be that part that could begin her journey and finding the rest of her family.
Are you still in touch with her?
I am, actually. I've actually gotten her in contact with my dad because, of course, her biological mother, come to find out, and my dad are first cousins. So he's actually been able to help her fill in some of the gaps as well. It's been quite an interesting side journey to this season of Relative Race.
Yeah, I can imagine. I mean, your lives have been, I would say, turned upside down, but that usually has a negative connotation. But I haven't seen anything negative come out of this. In fact, Deshay, you now fully understand your origins, which you never have in your life.
Yes, most definitely. You know, we're still in constant contact with everyone we've met on the race, my family and Chris's, and being able to foster these relationships and go out and see them after the race was awesome. I talk to my dad every single day.
Oh, wow.
There's not a day that goes by. And most of the Love family, you know, I talk to every single one of them at least weekly, I would say, and we've had get-togethers, and we're planning to have a end-of-the-season party together in Texas, so we're all real excited.
Oh, how fun. I mean, you're a perfect fit to that family, right?
Yes. They're exactly my personality, and they're so much like the family that I grew up with. To see that just instant, like I was just meant to be there with them.
Yes.
You know, I think everything in God's timing, and I just know that I was meant to meet them in this season of my life when, you know, our son Houston is 3, and so he'll never not remember a time where he didn't have his Papa Love, which is what he calls him. So it's just awesome.
Isn't that great? And so how has your adoptive family reacted to your experience and your journey with this?
They're over the moon excited. My mom, Cherie, and my dad, David, they are so supportive the whole time. My mom has actually been able to meet some of the Love family and just share the love. I mean, that's just how we all are. We all just kind of blended together perfectly. It's fantastic.
Well, it's Relative Race. It's on Sunday nights at 8 Eastern, 5 Pacific time. You can stream it using the BYU TV app. You can watch it on BYUtv.org. Chris and Deshay, Team Greens, thanks so much for sharing the experience. It's been really fun to watch.
Thanks for having us.
Thank you so much.
David is back for Ask Us Anything next. And David, this is a great question we got from Penelope in Ann Arbor, Michigan. She has emailed us at askusananything@extremegenes.com. Guys, I recently inherited a huge cache of family documents, 19th century to early 20th century. So excited. What's the best way to keep them preserved? Thanks, Penelope.
Well, first off, I would tell her what not to do with them. Like, remember those 1960s and 70s magnetic photo albums where you stick them down?
Yeah, the worst.
Oh, yeah.
And then there's paper clips and staples and
staples.
Tape.
Scotch tape.
Tape them somewhere. You don't do any of that. Don't do any of that.
Yeah, duct tape works for everything, not on family manuscripts.
All right, seriously, Penelope, because I know this is serious business, and you're absolutely right. You want to make sure that they get preserved and passed on to the next generation or to the archive you will eventually give them to if you can't find someone who is going to be as dedicated to keeping them as you are.
And one of the things that I do is I personally use Gaylord.com, which has archival supplies from document boxes to acid-free folders, non-PVC plastic sheets, because you want to make sure that if they've lasted 100, 150, 200 years, you want them to last more than just a couple of years in something that's not suitable for them.
Yeah, absolutely.
And don't frame them and put them in direct sunlight. Make copies of them. You can make good color copies. You know, it's going to trick the eye of any family guest. The last thing you want to do is plop them up on the wall.
Yeah, you're absolutely right. And I've had a lot of things that when I didn't know any better when I was young, I framed them, and they've faded quite a bit. Not gone, but still, I did digitize and actually clean up using Photoshop family Bible pages. And they're framed in my hallway, and they look great, and I look at them all the time. And once in a while, I'll crack open the book and look at the original in those acid-free sleeves. But that's all great advice, David.
Yeah, and you might find a conservator, like I did for the family Bible that was given to me earlier this year. The cover was falling off. Well, I don't want to use duct tape and repair it. So you want to find a conservator that might be in the area that can do that. And, of course, there are so many specialists out there that can take and remove mold and, you know, repair documents so seamlessly you'd never know. It's almost like plastic surgery of our family ephemera.
And, you know, sometimes things are just falling apart, Penelope. And in fact, I've had some old newspapers as part of my collection of historic stuff. I've gone out to places like, I don't know if you have a Benyon Craft in your area, but they have the means to actually relax old paper and then attach it using acid-free adhesive to foam core. And then you can use that to keep it safer because it won't fall apart anymore. It won't be flaking off on you. And, so, you know, sometimes they're just too delicate even to put into sleeves.
That is very true. Treat the past like you would treat a newborn.
Yeah, absolutely right. Congratulations, though. I know what a thrill that is to get a stash of family documents like that. 5 years ago, I had one of those big ones as well, and I know it'll never be topped. And it's such a great responsibility as well to make sure that those things are kept safe. Hey, don't forget also to make sure that you keep this away from heat ducts, air conditioning ducts, because when it gets cold and gets hot, it expands and contracts the paper, and that can cause it to fall apart. And keep it off the floor, too, in case there's ever any kind of flood. If you could keep it in a cabinet that's maybe just off the floor, that might be a great way to protect it that way. There's so many things to think about. Bugs, right?
Yeah. Well, and the other thing is make copies. Make color scans and distribute them for the holidays.
That's Ask Us Anything for this week. Thank you so much, David, and thank you so much, Penelope. You can always ask us any question at AskUsAnything@ExtremeGenes.com. Hey, that's our show for this week. Thank you so much for joining us, and thanks to Paul Woodbury for coming on and talking about spit to screen. What happens to your saliva after you send it in to the DNA testing companies? Hey, we'll talk to you again next week. And remember, as far as everyone knows, we're a nice, normal family.
This has been Extreme Genes. Share your family story by going to FamilySearch.org.